Welcome to the 50th anniversary of the Smithsonian Conservation Biology Institute. We're pleased that you all could make it and are very excited. My name is Peter Leimgruber. I'm the Director of Conservation and Science, and I have the honor to lead you through the morning. I would like to start with asking Brandie Smith, the director, the John and Adrienne Moss director of the zoo, to come up and give talks. Just a bit of background on Brandie. Brandie is a zoo lifer, I would say. She started her career as an intern. She actually was an intern at some point here, a curatorial intern at NZCBI, like many of the people in this room that started somewhere as an intern and then came back. She has worked for, I think, nearly a decade with AZA, leading various important conservation efforts, being the vice president for field conservation or for conservation programs. And then she came to the zoo in 2008 and was the general curator, the director of animal care. I get the titles wrong. You know, she was the big guy for the animal collection, and then eventually became director and has been leading the zoo since 2021. I have to say I'm really excited about the opportunity that she has created in terms of bringing conservation science, bringing the hidden zoo more to the forefront. So with this, please welcome Brandie.
Thank you, Peter. Good morning, everyone. Welcome to the NZCBI 50th anniversary celebration. You guys are a super mellow crowd right now, so I'm really, really excited about this. So we got to bring the energy up a little bit. So the first thing I want to do is thank everyone. There's so many people that it took to make today possible. So first of all, there was a committee, a 50th anniversary committee, who put all of these things together and really envisioned what today could be. So a group of hardworking people who are at the center of all of this. But also every single department was involved, right? It was conservation and science and animal care and IT and events and advancement and OFMR. And I know I'm going to miss a department. So have I missed any of your departments? What departments have I missed? Finance and admin, right? So every single department has been finance and admin, just the people who supplied all of the money to make this happen. And all of the people. So I really want to thank everybody because it took a lot of work to make today happen. And I also want to thank the board members. Our board chair, Ken Laurie, is here in the audience today. So the board members who are here today, the donors, the supporters, you know, everyone who loves us so much and who helps us keep going. So thank you. Peter mentioned that I was an intern here. It was actually back in 1994. I was a curatorial intern. Most people didn't know what that was because out of the packs of interns that ran around here, I was just one. But mostly it meant that I did whatever Larry Collins told me to do. And I also spent a lot of time in the small animal facility. And I worked with I worked with a lot of animals there. And I'll never forget my first day. It was Greg Peterson introduced me to tiger qualls. And he said, this is a tiger quall. It's basically a land shark, so don't let it bite you. And so that was when kind of safety and protocols didn't exist so much back then. So Peter, when you mentioned the location, the first aid kit, I had a little flashback, but it's all good. But I do remember kind of three things from that time. First of all, this place, right? The incredible beauty of this place and this location. It's just such an absolutely stunning place. You wake up every morning and you're grateful to be alive because you get to be here. I remember the people, and it was the people who work here. It was the staff, it was the students, it was the interns, just to be surrounded by so much energy and inspiration every single day. My research background is actually in population genetics. So, you know, Smithsonian's National Zoo and Conservation Biology Institute, Kathy Rawls, John Ballou, Rob Fleischer, right? The gods of population biology and genomics were here. So I was just kind of giddy every single day that I got to work in the same place where they were. And the other thing I would think, and I kind of thought it was far-fetched because I realized that once people come here, they never leave. But I thought maybe, just maybe, if I'm lucky enough, someone will retire I didn't think hit by a bus. I thought win the lottery, someone will leave or retire, and there'll be a space that'll be open, and I'll have the opportunity to work here. And so the fact that I am the John and Adrienne Mars Director of Smithsonian's National Zoo and Conservation Biology Institute, I can't tell you how deeply honored I am to have that title and to be here at this time. I'm deeply honored and quite frankly a little stunned, but I just want you to know how much it means to me to be able to represent this position at this time and be here with all of you. So the existence of this campus started about 50 years ago with an act of audacity and vision by Dr. Ted Reed. And I know that he was looking for a place for breeding and studying endangered species. And I think this is true to the story. There were a number of locations, and one was the Dismal Swamp. Was that it? I feel like the Dismal Swamp was in the running for the location for this facility. So I'm really pretty glad that this place was chosen for many reasons. But although it was originally founded to breed endangered species, it's become so much more. It is really, it's the nexus, it's the hub of all of NZCBI's conservation and science work. Like, I feel like in its sheer size and complexity, it's the physical embodiment of all that we do. I don't want to miss things. We do science and conservation in more than 40 countries around the world. We've trained over 2,000 conservation and science leaders worldwide. We're a living laboratory, tracking turtles, nesting kestrels, plotting biodiversity, all of these different things. We are literally a working landscape with hay and bamboo and bees and chestnuts and solar fields and water wells and all of these things. We're the home base for restoring ecosystems like Virginia Working Landscapes and our Great Plains Science Program and our bird-friendly coffee. And we partner with everyone, right? I think that's kind of the hallmark of what we do as scientists, but it's also embodied here with George Mason, with Neon, with Forest Geo, these active partnerships that keep this place going. And yes, we are still about breeding and studying endangered species. And through these breeding programs, our lab work, our field work, we save species from extinction. I wasn't going to say this, but I saw Pierre and Jesus in the audience, and I'm just going to say this. Like, there are some big, might I say, colossal organizations that claim that they are going to de-extinct species. It is literally what we do every day. You do that before you have breakfast. So this is something that we do. There are species that exist in the wild and on the planet because of the work of the people in this room and the people that came before us. So all of these species, golden-eyed tamarins, black-footed ferrets, giant pandas, Panamanian golden frogs, sahex, pea horses. I was actually thinking, is Annalisa here? Is anybody from communications here? Oh, there you are. So I was actually thinking, as I was like going through the list of species, I was thinking about Persian onagers, and I feel like we should get a T-shirt that says like breeding asses and taking names, because that's what we do. And even if we just make one and give it to Buddha, I think we're good. All right. And what we do, we're unique. We do things that only we can do. There's no one else. It's like, we talk about, are we conservation, or are we zoo? What do we do? It's the animals, it's the labs, it's the field, but it's all of those. Like, I'm old enough to remember that old, it's a Reese's peanut butter cup commercial, right? You got chocolate in my peanut butter. You got peanut butter in my chocolate. It's a Reese's peanut butter cup. Outside of a peanut M&M, it's the best thing in the world. So this is what we are. We have the animals and the lab and the field and the public. People are clamoring to come here and see what we do. Peter talks very, very eloquently about how we're the one-stop shop for de-extincting species. Well, not de-extincting, I know you hate that term. We are the one-stop shop for saving species. Good? Okay, all right. He's very eloquent. I will let him say that when he speaks. But we're unique in what we do, and we're unique in who we are. I was at the AZA conference last week, and they give out the Deborah Kleinman Scientific Advancement Award. We, Deborah, our scientist, is synonymous for scientific excellence in the entire zoological community, right? Her name, our place, is associated with that excellence. And when I give tours and I talk to people, the things I tell them about the firsts, right? There's so many places that we're the first, we're the best, we crack the code, right? Devra and Golden Line Tamarins. Kenton, if you didn't watch the last All Staff, you have to watch it. There were great presentations by everyone, but Kenton was talking about Devra and about her impact on Golden Line Tamarins, and the fact that if she didn't exist, there's a big chance that there wouldn't be tamarins in the wild. And what she did is something that we do in terms of cooperating. She put together collaborative teams to save species, right? Another thing that we do so well, these collaborative teams. There's Joe Gale, her AI on felids and on giant pandas. Janine and Steve and Rob, the work that they've done with non-invasive monitoring for endocrine and genetics, not just the impact it has on our ability to collect information, but the impact it has on the animals themselves in promoting this incredible positive welfare. The entire pathologic path team discovering EEHV, and I'll just mention them again, John Ballou and Kathy Rawls, my kind of personal heroes given what I do, right? The gods of population biology worked here. How we genetically manage endangered species in small populations was developed here by individuals here. So as we're thinking about this, I do want to celebrate the past. I want to revel in the moment, in the present, but also let's take this time here, this day here, to be visionary about the future, right? The audacity and the vision that brought NZCBI to life. That's what I want to see today. I need you all to give me these stories of the firsts and these groundbreaking moments when we, our scientists, our science, made the world a better place and positively impacted biodiversity across the planet. So I'm looking forward to today. No pressure. But in closing, I will introduce Dr. Ellen Stofan. She is not here today because she's fighting the fight at Climate Week in New York. But we're going to have a video of her she wanted to be here at least virtually, and she will be here at the dinner tomorrow night. But I'll hand this off to Ellen. Thank you.
Hi, I'm Ellen Stofan, the Undersecretary for Science and Research at the Smithsonian. This year marks the 50th anniversary of the Smithsonian Conservation Biology Institute. SCBI has led the way in conservation action by training thousands of scientists, leading the recovery of endangered species, and pioneering research that shapes how we care for wildlife and ecosystems around the world. 50 years of dedication to fostering resilience, advancing conservation, and sparking innovative solutions for people and nature. This is the Smithsonian at its best, creating and sharing knowledge to uplift communities everywhere. Thank you for being an essential part of this shared journey. Here's to the future we are building together.
I'm guessing some of the SCBI alumni in the room are wondering, who is that guy? Where's he from? I don't remember him. In truth, I've only been here for about a year and a half, but as Peter mentioned, I've been in the zoo world for about 35 years. First time I was out here was in 1992 for a workshop on population genetics, because that's my background as well. I have come back numerous times since then and seen a lot of the change. So maybe that's why I'm up here. Anyway, we're going to go way back in history. Not that direction. This way. Many of you know this picture. We have it on a graphic at the zoo. This is William Temple Hornaday. By many, he is considered the founder of wildlife conservation in America. Not conservation writ large, like national parks and whatnot, but wildlife conservation. He was the first director of the National Zoo. He was not for very long, but he was. He is an important part of our history because he brought animals down to the Capitol. He was the nation's taxidermist. Yes, that position existed. He was out collecting animals. He was a big game hunter, and he saw that a lot of populations were vanishing. The word endangered wasn't used back then. They were vanishing animals, all right? He brought 15 different species to the Capitol Mall, bringing ambassadors, probably for the first time, ambassadors of the species rather than ambassadors of the country they were from. And he wanted to show people these are animals we could lose. And he wanted to start a zoo. Bison were one of them, and that's the picture everyone knows. As I was looking into this, do you know that he brought black-footed ferrets too? Back in 1888, we can start our history with black-footed ferrets. Anyway, moving on. All zoos, I think nowadays, use their animal collection as ambassadors for the species themselves, to talk about their status in the wild, how we can help, things like that. But zoos have gone well beyond just being places for ambassador animals. Starting very early on, zoos started purposefully collecting animals, breeding them up, and then releasing them back into the wild. The first example I could find was in 1907, 15 bison who were at the Bronx Zoo. From a herd there, these were surplus, were sent to protected habitat in Oklahoma. Astounding, I think, back in 1907. 1902, a number of alpine ibex were collected. This was over in Switzerland, also a vanishing species. Those animals were brought to two zoos in Switzerland. They were bred up until there was a herd and there was surplus, and they were released. Right? Next one I could find, 1927, the European bison or viscent was considered extinct in the wild. There were a few left in zoos after World War I, surprisingly. There were a few left. A pair were put back in a forest in Poland, and that population still persists, and I hope they put in more after the first two. But anyway, moving on, in 1980, Arabian oryx that were extinct in the wild were bred up mainly at the Phoenix Zoo, also San Diego Zoo got involved in that, and they were returned to the wild. This is what zoos were able to do, right? Zoos weren't the only ones. Peregrine falcons were released starting in about 1974, but that wasn't done through zoos, right? North American river otters were extirpated from large amounts of their range, and they were being put back with translocation. So I'm talking about zoo-based stuff. I found it interesting that in the history of the National Zoo, we have had a total of 15 bison. Yet back in 1907, the Bronx had a herd, they bred them up, and they had 15 that they could send back to start repopulating the wild. All of these animals, these four, have hooves and take up quite a bit of space, all right? The one thing that we needed to really become players in the business of restoring species was space. And 50 years ago, we got that space. Amazing. It makes us players. Okay, quickly, I'm not going to go through any of these individually. We've had thousands of animals out here, lots of hoofstock, but lots of other things. Tiger quolls, for example, and tree kangaroos, all sorts of things, all right? And it's not just mammals. Thousands of birds have been produced at this facility. And I get all these records from the ZIMS database, so I'm assuming they're all correct. Both waterfowl, some people know me, and non-waterfowl, tremendous numbers of animals have been produced here. This is today's collection, and you can see that almost all of the species up there are either endangered in the wild or vulnerable based on IUCN criteria. These are species that we are currently working with. I went through all of those numbers quickly for a reason. We're going to celebrate conservation and science today, yes. But we have to celebrate the people who took care of those thousands of animals. They don't just get along by themselves, right? It takes staff, it takes facilities, it takes time, and it takes expertise that has grown right here. There are species that were not bred elsewhere until they were brought here and the work was done here. It's not just about science. It's not just about reintroduction. It's also about basic animal husbandry. It was worked out at this facility by over the 50 years, hundreds of people dedicated to professional animal care. I don't want people to forget that the animal collection enables a lot of the science, enables a lot of the conservation, and there are people here who take care of those animals. All right, this it's already been said pretty much. Just taking animals and putting them back, it's not that simple. It might have been that simple in 1907 with bison because bison could be treated like cows, right? They were a lot like cattle. We didn't have to do a lot to figure out how to breed them. Not the case with most of the species we work with today, all right? The zoo side is to produce healthy animals, surplus, so that we can reintroduce them. In order to get there, there's a whole bunch of science and basic husbandry that has to be worked out. I listed just a few of them. Their physical needs, dietary, social. Very often we need to know genetics because a healthy surplus is only produced by a well-managed population, as an example. So the reproductive biology of the species, all sorts of things need to be in place in order to have a successful population. And when you're releasing animals, you don't just say, be free, right? You've got to track them. You've got to make sure what you're doing is effective, that you have produced animals that can thrive in that environment and can be restored. So the point of this talk is to say, we have the space, we have the research, and that's what makes us an outstanding and effective conservation organization. Thank you.
Good morning, everyone. And yeah, in a lot of ways, coming here feels like returning home. And like Brandie, I sort of have a pinch me moment to realize that I'm now in this position. And it really is my honor and a pleasure to be speaking to you all this morning about something that truly is near and dear to my heart, which is the relationship between SCBI, NZCBI, that still doesn't roll smoothly off my tongue, forgive me, and George Mason University. And this is a partnership. Oh, there we go. Okay, thank you. So this is a partnership that has come about to invest in the future of conservation through this flagship program, which is the Smithsonian-Mason School of Conservation. And this is not simply a marriage of convenience due to proximity. Okay, this is the joining of two institutions that have a common vision for a world where conservation literate people integrate species and habitat conservation into global efforts and outcomes. So SMSC programs capitalize on the significant strengths of each institution. As you all know, NZCBI plays a leading role in Smithsonian's global efforts towards conservation by hosting the field, the renowned scientists who apply their extensive knowledge to conservation science, as we're going to hear all about today. But this place also has a long history in training wildlife conservation leaders, both here in the US and around the globe. What you may not know, though, is about George Mason quite as much. And we are leaders in innovative transdisciplinary education focused on trying to drive solutions to the grand challenges of our time. We also are known for an extensive portfolio of academic excellence in the STEM fields. We also are the largest university in Virginia, which surprises a lot of people given how young we are. And we are a top-tier research university, recently recognized by the National Science Foundation for being one of the top 10 public universities in the nation in research growth. And so together, we are developing leaders in biodiversity conservation through education, training, and research. And this relationship that we have is decades in the making. And it first started with training graduate professionals and graduate students. Borapat, who's here, and I were lucky to be among the first cohort of a joint PhD fellowship. And we've had a number of PhD fellows over the years, some of which are shown here, that all have become leaders in the field of conservation, whether that's in science, management, or policy. And then thanks to the vision of Steve Montfort and Tom Wood, both Mason alum, by the way, we expanded our education portfolio in 2008 to include a unique semester away program where students lived and learned here in the training center for 16 weeks. And it feels like in a blink, the main campus that a lot of us here probably remember, with the corn silos and the waterfowl habitat, transformed into what we're in now, which is this beautiful LEED Gold certified facility that opened in 2012 and has allowed us to greatly expand our educational opportunities. And at the center of SMSC continues to be our hallmark program, which is the semester away programs. Here we have classes led by SMSC faculty who work to incorporate hands-on learning and networking experiences for our students. And this includes working with researchers here at the Smithsonian and our vast array of community partners in the field of conservation. Currently, we have 3 semester away programs. These are all 16 credit immersive programs. The first of which I'll talk to you about is the Conservation, Biodiversity, and Society. Don't know why that advanced. But in this program, we're teaching the ways that science management and policy, hello. All right, hopefully this is not on a timer, we'll see. Bear with me. So our first program, maybe we are, shoot. All right, we're gonna see if we can preempt this. Where we combine science management and policy to address current conservation issues in and out of the field. Our WEC program teaches traditional and cutting-edge field techniques to survey species in the wild and to investigate ecological patterns and processes. And our semester that we offer in the spring, ESC, this focuses on small population biology and the challenges within those and the strategies that we use to try and help save these populations from extinction. Our students are in the class 5 days a week, class being either in a classroom or out in the field doing field work, and often these days are extended with special field trips and other opportunities. They are fully immersed here in the SCBI community, being able to participate in special events like this one we're having today. And they get to interact with the renowned conservation leaders who come to this place. And for instance, 3 years in a row, they were lucky enough to be able to interact with Jane Goodall. Depending which semester program they're in, they either do a 1-day practicum, 1 day a week, or they do a 5-week, I'm stumbling over my slide, sorry, or do a 5-week research placement. These give our students opportunities to get hands-on current conservation skills that they can carry on into their future careers. And the vast majority of our students do come from Mason. And we hear from them time and time again that they enrolled at Mason because of this special partnership we have with the Smithsonian. But we also have 6 consortium schools where we have special partnerships for their students to come. And the reality is that any student at any college or university can apply to attend as well. Now, to me, the outcomes for our students speak volumes about the quality of education that they're getting here. This is just an example of some of the graduate programs that our students have gone on to attend, whether that's studying science, veterinary medicine, or even law related to conservation-related aspects. We've had 2 students go on to become faculty at Penn State University and Washington State University. We also have students who have gone on to work in notable federal, state, and even local governmental organizations, as well as an array of non-governmental organizations, all focused on conservation. You shouldn't be surprised. A large number of our students have found employment through NZCBI. I think we're in the double digits easily at this point. But we also have students placed at zoological facilities around the country. So just like our PhD fellows, our graduates of the semester away program are making their mark in science, in management, and in policy of conservation. Now, forgive me, this is, for whatever reason, the place in my talk where I seem to switch into late night infomercial mode, as I'm thinking, but wait, there's more. So the semester away program is not the only thing we offer. We offer summer classes as well to our undergraduate students. A number of them are shown here. These build upon the skills that we teach in the semester program, as well as providing an opportunity for undergraduates to come and learn that cannot afford, for whatever reason, to participate in one of our 16-week programs. We have incredibly popular high school programming in the summer as well. These classes fill up fast and teach skills such as field skills and natural history illustration. And of course, building off the long history that this place has in training professionals, we are offering graduate and professional courses. These may be 1 week or 2 week immersive programs with people living here, or these days it's virtual offerings as well. And these courses are designed to fill a critical gap in capacity building of high demand skills for researchers and practitioners in the field of conservation. Examples of some of these, we have an array of classes teaching people the skills they need to use R for various areas of conservation. And this is where I get to say, I'm not just the executive director, I'm a customer as well, because I've taken one of these classes and I've sent all of my graduate students to these classes. We also have courses in things like camera trapping, conservation genomics, zoo nutrition. And so though this partnership is decades in the making, I really feel like we've only just begun because we are ripe with opportunity. And just some of the ways that we are moving forward, we now have shared tenure faculty position. We've launched a new BS in integrative conservation. And we have the world-renowned endocrine laboratory that currently is housed within SMSC facility or buildings, just temporarily, I hear. We have an exciting new grad professional course that's being launched in January, and this class is being co-taught by faculty from MIT and the UK National Oceanographic Conservation Center. And for a select lucky 10 undergraduates this summer, they will get to come and live here for 10 weeks, study with our amazing scientists in our first ever NSF research experience for undergraduates. We are looking to expand our consortium partnerships and to reignite that PhD fellowship program that myself and Borapot and others have been able to take advantage of. All of this, of course, would not be possible without the incredible, talented faculty that we have, our amazing team behind the scenes, and of course, the numerous staff and scientists, both here at SCBI and at the zoo, who contribute not only their time, but honestly their heart to this program.
So in 1974, as I said already, this place was acquired and opened in 1975. It really only represented a milestone in the history, in the accomplishments, in the evolution of the National Zoo into a global conservation organization, but an important milestone. So the program started way earlier, but this really became, as Brandie said, the physical embodiment. It was a place where the science could happen away from the public and where we could really start to innovate, prototype, test, do research, and then eventually also develop tools that we could take out to the wild. I spend a lot of time thinking, well, what are the highlights that I could, in a brief talk, bring up about our history? And there's just so much. This is the first 25 years and just a few things that came to my mind, really starting very early on with really revolutionary new field programs in the Smithsonian Ceylon program, for example, in 1967, the Smithsonian Nepal Tiger program, which was followed with some rhino work. Eric Dinnerstein is here. He's been to this place. And innovations like Kathy Rawl and John Ballou really determining that inbreeding has detrimental effects, who knew, on the survival of populations, small populations. And I could go on and on. And I'm sure there's quite a number that I'm missing. There are probably people in the audience that are going like, where's my thing? And so I apologize for that. And it goes on. I mean, there are these things, for example, camera trapping. Bill McShay and his research efforts was at the forefront of figuring out how to use camera traps to monitor biodiversity. He helped create eMammal and then Wildlife Insights, and now he's working on Snapshot USA. You know, we were part of the reintroduction of the Simatow oryx, which is one of the species that went from extinct in the wild to endangered just 2 years ago. It was downlisted. So there are all these successes. The question is, how do we build now from this place where we are, and what do we do in the future? And of course, we're not going to start from anew, right? So we're going to build on our successes from the past. We're going to build on our expertise, our infrastructure, our resources, and we try to figure out how do we better align that to find the next cutting edge thing to do. One thing that strikes me that I hear again and again, I hear it from our board, I hear it from the public, is we're a hidden zoo. Our partners know it, the academic community knows it, that we're here and that we're doing this work, but the public doesn't know it. And that has an effect on our resources. If people don't know what we're doing, they can't come and support us. So some of it is really about us to kind of step back and think about how are we communicating about our science? Are we communicating about it based on our departments? Or are we communicating about it based on themes that actually are compelling and that bring the public to care about what we do and help us support? And if I think about the 4 themes that pretty much encompass almost everything that we do, then for me, it's something on restoring species, because we do it from working on the genetics and genomics of the species to the reproductive biology, all the way to figuring out, are they surviving in the wild? Can we track them? Can we work with the communities to make sure they actually are not being poached? And we should focus on that. We should build that out, and we should call it what it is, rather than talking about, oh, okay, we were able to build new artificial assisted reproductive techniques for this species. That's important, but that's a minute detail when we're about to save an entire species. Not a minute detail for you and for our funding, because it's critical, but the public really cares about the outcome. Similarly, I think we are doing critical science here, understanding the ecology of wildlife. Russ Greenberg, many years ago, he was the head of the Migratory Birds Center. He called it the ecology of extinction. Scott Yanko, who is a new staff member, has a new proposal in it. It's the ecology of death, I think is what he calls it. So ultimately, it's really about who, where, why, and when is out there. What animal is out there? Why is it there? And why is it dying? Or why is it surviving? And we collect this data, and we need to mobilize it. We need to give it to the public. I'll talk more about this. Beyond 3030, 30 by 30 refers to the conventional biological diversity commitment to preserve 30% of land and water by 2030. That's great, we support that, but what about the other 70%? And we are working on this other 70% with our working land and seascape, so we need to talk about this. And then global health. We're actually struggling to formulate what global health means to us, which is crazy to me, because we have a concentration of veterinarians, of animal physiologists, of wildlife biologists. We need to lean into this topic and figure out what does it mean and how do we actually implement it on the ground. Brandie referred to the big ones in the room, Colossal, there's Revive and Restore, there's Rewild. There are many organizations out there that we can look at that do this really well. Colossal has raised $550 million. It's a business valued at more than $1 billion. We would never do what they do. That's not what we do. So I'm not trying to say that. What they do is, well, they're using the idea of de-extinction to raise money for technologies that then they hope can have applications in the human medical field. We're not going to do that. But the truth is, we need to have some of these biotechnologies, and we can be at the cutting edge of developing some of them. Similarly, I mean, I think Revive and Restore and Rewild are great NGOs, but they're really doing a very specific part of this whole issue of restoring species. And we're kind of across the entire spectrum. Oops, something happened to my formatting. You can still read it. So We are doing quite a bit of conservation omics, but I think we do need in the future expand into this. So this really means to perhaps create the capacity for translational genomics to be able to kind of work on the adaptation of species to their changing environment. But it also means leaning into the technologies that we're currently developing. I have this microfluidics slide here that Jennifer Nagashima made for me and that I love, but it is really complex, but you can see what it is. It's the idea of creating these organs on a chip to then improve the way we grow tissue and cells that we get from our biorepositories. So all these technologies are critical for what we're doing, and we're already doing them, but how much does the public know about it, and how much do we lean into that? Next, okay, I'm in control. Theoretically. I think one thing that to remind, so for me, Wild Futures, I talked a lot now about these biotechnologies. That's, of course, only one of the things. But we're already doing a lot of this other thing. It takes a lot more than the biotechnology to restore a species. But I do think there are a few places where we could do more work. I find it very compelling to look at using behavioral ecology to improve reintroductions. This is not new. Deborah Kleiman was kind of the pioneer in this, but there's still a lot of work to be done. We're now realizing that you can't take the average behavior of an animal to predict what will happen. There are animal personalities, especially true for socially evolved species like elephants. We need to kind of work on those tools and technologies and better understand them. So then I'm coming back to the tracking. We're already doing a lot of tracking. I mentioned Bill McShay with eMammal and Wildlife Insights and now Snapshot USA. We're using the camera trapping as a bit of a prototype to think about what does this mean for us and the Smithsonian. In the past, biodiversity information was physical specimens at a museum. We have the largest collection of physical specimens. I believe we're the largest contributor to GBIF, which is a global database on biodiversity based on our collections. But the future is not in physical collections. The future is going to be in digital data. And we're already doing it. And so we're now talking about how do we create digital biorepositories at the Smithsonian. Seems like we should have them already. But the truth is, all this data sits in the Google Cloud. If Google decides to put a paywall there or to not do it anymore or to charge us for it, which would be millions of dollars, it could just go away. So we really need to develop that safe space where we as an institution can provide this data, have it for analysis, and show others how to do it. So we're going to do this with Snapshot. We actually have funding to do this for the next few years to develop this prototype of a digital biorepository. We need to accelerate our use of technology. We have workshops here last year that talked about how to better integrate across NGOs and different technology types. Everyone talks about AI, right? If you bring a problem, someone says, let AI solve it. Can't solve everything, but there are places where it would be really useful. This is a paper co-published by one of our scientists, Jared Stabach, that uses AI to automatically detect and map migrating wildebeest in the Masai Mara Serengeti ecosystem. So it can be done, especially in open landscapes. Other things here would be environmental DNA. Everyone wants to do environmental DNA, but it's not really clear how well it works. And there's a lot of space there for us to lean in and do the foundational science for it. Ultimately, right now, we're pretty good at detecting individuals. We're pretty good at tracking individuals, but we're not great at going from individuals to populations, what is actually happening to these populations. And we suck at saying anything about are these populations declining or going up? What are the demographics? You would think at this stage we would be much better, but that's an area we need to do a lot more. And I hope that will come. Beyond 30 by 30, I don't know how many of you are familiar with our programs. We have many landscape-based programs. In the past, we combined them under the Working Land and Seascapes initiative across all of the Smithsonian. That went away. We're going to bring that back. And we're going to integrate all of our landscapes into that. And we're going to try to replicate these approaches that are looking at how can we work with local landowners local communities and local people to actually implement conservation. What are the sustainability solutions we can develop, and how can we roll them out? We do quite a bit of this already, but there's space to kind of generalize that. And that requires social science. We don't have a social scientist on staff. I'm not sure we will ever have the resources to do that. So who is the best organization out there for us to be partnering with? That's the other piece. We don't need to do it all by ourselves, but we need to be more strategic about finding big partners that can help us accomplish some of these things. I think social science is one of them. There are many really good schools out there. Just coming back to this 30 by 30 versus 70%, right? Protected areas don't look like this, but this is the land, I think, where we have an opportunity to preserve a lot of wildlife. And we need to figure out how we can harmonize human land use that's economical with the need for conservation, and whether that is figuring out how we deal with linear transportation infrastructure, or whether that is sustainable management of the existing lands. Global health. We do quite a bit already. We actually have a global health program. It's mostly focused on Kenya. The basic ideas behind it are there. We have many veterinarians, many experts in wildlife health, in conservation medicine. But, you know, and we do a lot, but it's all kind of a project here, a project there, a project this. And we need to kind of figure out what are the main questions there that we want to address. Part of that could be solving or mitigating zoonotic disease or specific disease issues. Can we solve the problem of chytrid fungus? Can we address the issue of yellow fever killing primates in South America as climate change has it spread from the Amazon into other regions? And then James Hassel, who unfortunately couldn't make it today and leads our program in Kenya, he has a new initiative that's called Ecosystem Health and Finance, which is really looking at funding opportunities where we better link ecosystem health with public health. Can we demonstrate how ecosystem health affects public health and then tap into this funding source for public health? So those were a couple of ideas. I know that they're perhaps a little far out, but I think what is the right stretch for us to go? What can we achieve? And what is the right challenge to get there? Before I end, we don't have much time to talk about all the incredible people that are here. So I thought this is not a complete picture with everyone that's there, but you might recognize some of them. It includes former directors, Chris Wemmer, Steve Monfort, John Eisenberg, and many others that you recognize, Deva Kleiman, and there are many missing. I hope some of them are here some of our alumni are here in the room. As Brandie mentioned, these guys are giants in the conservation world, all of them. But behind them was always an army, a community of people that have supported this work. I wish we had more pictures of this. These are pictures I found from an old video that Miles made about the Conservation Biology Institute. It also shows a little bit of this, we call it residential learning community. It was just a fun place to be. You know, you play volleyball at night, you work together with others in, you know, on animal care or on science, and, you know, and you build your personal relationships and your professional networks for the future. So for me, it's been an incredible place to be, and I'm very grateful that I had this opportunity. And I love this picture, and I want to say thank you with a greeting from Joe Gale.